I recently had a deep, profound discussion with Joachim Gerlach. He's the man who is focused on the spike protein, one of those rare researchers and scientists who took this and has just kept on hammering home to understand what is going on from a pathophysiology point of view and critically to try and find solutions. We had a detailed conversation, almost 2 hours of content, looking through slides, discussing the implications around spike protein. And the point is that it seems that detox is a critical part. Many people would have heard that concept and they think, oh, come on, what are you talking about? Well, let's look at the science, and this is what we'll be looking at. What has he found? And critically, what can be done about the spike protein if it's present? So these are some important discussions, and we are, I think, having the first detailed discussion like this that you will ever hear. So without further ado, let's bring back in Joachim. We had a long discussion the last time, Joachim, but just in case somebody is jumping in at this point, just a quick introduction to who you are and what you're doing.
Yes, good evening, everybody. Good evening, Philip. Thank you for having me on. My name is Joachim Gerlach. I'm from Germany, and I'm a researcher and developer for dietary supplements, and I'm working in a large collaborative with a lot of scientists globally in different laboratories. I'm financing a lot of research with my company and my partners, and we are trying to mitigate the problems that come from the S protein and from also the virus itself.
So this is some big stuff. So as I said, we spoke for almost 2 hours about the spike protein. And let's start at this point, because I think this was an important point for people to try and understand. What is this talking about?
Okay, we are talking about persistent spike, persistent virus, and the condition it brings cells into. And so I chose the English word of I say the poetry, like the one rotten apple in a basket can spoil them all. And so that is resembling the cells that were invaded or are producing spike proteins or are still harboring viral debris or virus itself. And then on the right side, you see fried eggs, which resemble the conglomerates of cells that are formed when they get into contact with a spike protein and or the virus. That is a pretty common thing in also other viral infections, and it serves as a hideout for the spike protein and or virus. And so that's why I chose these two analogies to make it more visible for the listener and watcher.
I think that this is important. So in effect, even though people may be asymptomatic, it's likely to be in a lot of people a hiding place where spike protein is just sitting and potentially causing long-term illness. I've come to the conclusion when I look at patients who have long COVID-like symptoms or post-COVID, I suspect that they may be just more sensitive to the presence of the spike protein and they have symptoms. Would you say that a lot of people may have the same presence of spike protein and be asymptomatic, but not necessarily not at risk from the complications from it?
I mean, we see a lot of results coming out of the lab from patients that are completely asymptomatic, so to speak, but they harbor a lot of spike protein intracellular in the immune cells or in other cells. And so it does not necessarily reflect in symptoms. And I would be very careful in long COVID anyhow to rely on symptoms, because we have seen a lot of brain imaging, for example, where you see that there is already ongoing brain damage and these people are completely asymptomatic. So symptoms is a subjective thing sometimes. In many other diseases, maybe it is very helpful, and I think also in long COVID, but I would always rely more on visual or other diagnostics to see what's going on.
So let's go in. So where are these spike proteins hiding? Where are they sitting?
Yeah, so we'll dive into that. First of all, we have to understand that there is not one spike protein. First of all, there are many variants, and even variants in the vaccine. So you will not see that one prototype spike protein across the board. There will be the Wuhan, there will be the Delta, there will be Omicron, different Omicron variants, etc., etc. So spike can come from the virus. Even in a viral infection, a lot of spike is cleaved off, is cleaved and then into S1 subunit and S2 subunit and can circulate. So I colored them in blue because later we're going to have more slides so that you can distinguish. And then the spike from the vaccine, I put that in red. So that's a lipid nanoparticle in red above that. And that is transporting RNA into the cell and then converts the cell into a factory to produce spike proteins, which will be different from the virus. They have a different structure. And so they are maybe not, it's a big discussion. Are they more dangerous than the ones from the virus or not? But there, I put them in red. And then we have something that most people are not aware of, and even we are still struggling to resolve that, is the spike from bacteria, which Carlo Brogna has discovered that the virus can breed in our bacteria. And we find that spike protein that was produced within the bacteria has a different proteomic profile than all the other spikes. And you can now detect it and quantify it. So that's really important for us now to see if we can mitigate that. So we have to distinguish between roughly 3 different kinds of spike, and that's why I put them in these 3 colors.
You know, the spike protein from the bacteria, there are a lot of people who don't even know that this is relevant. And I think that it's important for people to know this is part of the reason why there is so much persistence of viral protein in the gut, and also why they find it in the wastewater all the time, because it is just persistent there, especially in the gut linked with bacteria. This is a huge issue that very few in the scientific community, except say Carlo Brogna and yourselves, have been focused on. And it's, I think, probably the great oversight from the COVID pandemic.
Yeah, it makes it, of course, more dangerous that or less likely that this virus will ever disappear, because now it has a huge reservoir, even in animals. I mean, you can see studies that they find virus and spike protein in cockroaches. Of course, they can't infect the cockroach, so it's most likely it was in bacteria that is in the cockroach that they could find then the virus. So it is also opening up the way, the path for zoonotic events, for different recombinants to occur, etc., etc. Plus, I mean, the implications for our gut biome and the dysbiosis are tremendous, inflammation, toxin-like peptides. So there's a lot of things we can make just a few hours on that alone. But
Yeah.
it's a source for spike protein, and we find it, and we have to deal with it. So where can these proteins be detected with conventional, now available diagnostic or research diagnostic tools? First of all, everybody might think, oh, we look for free spike in blood serum samples. You would find all 3 variants there. But it's not very successful. We work with the labs that do this, and they don't find much free floating in the blood. It's because of the nature. The spike protein is very sticky. It will not float around for a long time unless you have a production ongoing of spike and or virus or whatever. Then we find spike in PBMCs, peripheral blood mononuclear cells, which are immune cells, anything from macrophages, monocytes, T cells, CD4, CD8 lymphocytes, white blood cells, so to speak, immune cells. They can all harbor spike protein from all 3 sources, and they turn also into a very inflammatory and immune dysregulated state. Then you find spike in the blood and serum floating around in exosomes. That is very common. That is normally a sign of a viral persistence in some cells. Very low grade, but it packs and ships spike.
Can I just clarify here in case people are watching? So the exosomes, these are like the packages that you're sending to families and friends. So cells package different proteins, RNA, sometimes DNA, and they send them off to be picked up by other cells. And so what we seem to be seeing is immune cells packaging these spike proteins and the like to be sent elsewhere, probably because they can't deal with them.
Yes. And then you have spike in host cells that can be practically all organs are susceptible. I would say the big connecting part is, of course, the endothelium and the vascular system where spike can persist, but also in epithelial cells, in neurons, in liver cells, and even in stem cells. So that was just to show you the scope of the problem, where things are. You know?
So now you've brought up another point, which is that even though the spike may be present, it's not necessarily being circulated, because in some cases it's productive versus non-productive.
Yes. So the spike protein itself cannot reproduce. So when it goes into a cell, then you see what we see on the right side. You will have a cell with a persistent spike. It will enter, and it will also dysregulate all cell signaling and turn that cell into a senescent state. But it's not productive. But on the left side, you see the productive sources. Most of all, of course, cells that were transfected with a lipid nanoparticle and now got the RNA instruction to produce spike proteins. And then that's in red. And then in the middle, you see a virus persisting in a cell, also producing spike protein. And both of these cells can also excrete the spike protein in exosomes. That's why I put it there. And then you see on the bottom the bacteria, and also they can of course reproduce virus, but they can also probably excrete spike proteins because we find them in circulation. We don't know, it is still hypothetical, if they can also wrap them in extracellular kind of vesicles.
So we don't know if this is necessarily part of the picture.
I couldn't finish the slide, I forgot.
Yes.
Question marks.
So, yeah. So just to clarify then, when you are thinking of removing spike, you are thinking of taking it out of both the non-productive and the productive cells.
Exactly. Yes. Yes. So that's the idea, and you have to do it that way, because otherwise you will not succeed in mitigating the problem.
And so this is then leading us to the point you were making last time, which we'll come back to, that you need sometimes to stimulate cells to release the spike protein. What is this about?
It was, first of all, for diagnostic purposes or research purposes, you cannot rely just on taking blood samples from people and hoping to find all spike that is present. And so if you just take blood and you analyze it, and we have that on the left side of these bars, you have a very high blue bar, which means negative. So that means in the lab you couldn't detect any spike. And then these people are taking some capsules, and then 2 hours later, all of a sudden, the picture shifts quite dramatically. All of a sudden, you only have 15% positive and 85% being negative and 85% being positive. So that means we were able to mobilize spike out of the intercellular space into the extracellular space and into the bloodstream, so it can be detected. So now we have a better, let's say, a better accuracy in defining who has spike and who doesn't have spike.
And so what I would say, Joachim, is that I just lost you for a second there, but let me get this clear then. So your provocation that causes cells to release the spike protein, is that a normal thing that anybody can take, or is it a very specific thing that you guys have created for your testing?
It's now in the 4th generation. That means, look, you have different substances that the organism can use to selectively break open these cells. But as we have learned, the spike can be in so many different cells that you want to be sure that you have a good mix of molecules that will then trigger the release from all affected cell types. So you can't just say, oh, I'll take this molecule or that molecule, and that'll do it all. It will take then some of the cells out, but it is not really reliable. I like to be more accurate in how we proceed with this.
And so I think what I'm trying to get at is that this is not, the provocation is not a simple supplement that you can just get over the counter somewhere. This is something quite specific that you guys have created with your spike test.
Yes. It took a lot of work and a long time trying that.
Oh, yeah. So explain what this here is about with that provocation.
I really like that it came in a few days ago. So this is a different kind of diagnostic methodology. It's a mass spectrometry from our colleagues. And so you see here a spike provocation. You see we have anonymized this. So ID number 72 is before provocation. And then you see different columns. Spike glycoprotein 6.3, that is considered negative. Everything above 30 is positive. And then you see in the middle column, spike PP, double proline, that is the spike from the vaccine, not detected, ND. And then you see on the right, you see bacterial sequence that is not yet fully operational with all the values, but it gives you 1x means low. So there's a low presence of bacteria that was formed in the gut bacteria. So we can identify them. There's certain amino acid sequences in that spike that give it away that it is from you can distinguish them that it's from the bacteria, not from the virus or the vaccine. And then and I took this picture because it's a classic. After provocation, all of a sudden, the spike from the virus became positive with 33. That's a level when you consider that positive. And all of a sudden, in the middle, you see the spike from the vaccine shows up. Before, you couldn't detect it. So that was probably not from a productive process. It was it was still in the cells embedded, and the body can't get rid of it, and we provoked it to come out. So this patient does have some spike protein from the vaccine also persisting. And now we are really not sure yet what to make of the third row on the right. Why do we see now an increase in the spike from the bacteria when we are knocking on the door of our host cells? So again, still open for debate, open for research, another round, try to find out what's going on here. But this gives a clear picture of what to expect when you do this.
So that's critical. This is important stuff. So the provocation causes these cells to release whatever spike protein they have. And the practical question is it possible that they have spike protein left even after they have released some? Is it?
This is of course just a very light provocation. This is not what we would consider something like a detox. The dosages and the duration is way too low. It's just like provoking a bit. And you have to see these molecules. They don't go if you give them once at that dosage, they will not be able to penetrate into the whole organism and go everywhere. You will have a barrier-dependent cascade of how they function. You take them in, so the first part they will reach will be the gut epithelium. From there, they can go into the bloodstream. They might hit maybe first liver or some PBMCs or some endothelial cells. But they won't go into the bone marrow, not that fast and not at this kind of dosages.
Yeah. So this one here is what you found, similar principle before and after provocation.
Exactly. So you can clearly see here that there is a tendency, and we don't have hundreds and hundreds yet of reports, but you can see a tendency that this seems to be seems to be a good way to go. We should keep doing that now and document it.
So 2 hours, and these tests go from negative to positive. This is almost 1, 2, 3, 4 versus 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So almost 3 times the number that would become positive after provocation. Very interesting.
And that is also, we are entering now comparative trials with other companies and groups. And I'm trying to convince them now that we agree on a standardized testing. Yeah. Because if you imagine you don't provoke, then first of all, you have a lot of patients that seem to be negative. And then even when you did a detox and maybe helped a little bit, you still don't know, have you removed that from the cells, from the intracellular space? You will lack that information. So we need to include that into trials and try to standardize the way we look at patient and report about outcomes.
Excellent. So then what is this about? Because so far the provocation is talking about, is it Xenolytics or Xenomorphics or both?
Yeah, so this is very important because I was at a conference in Munich the other week, and a doctor came to me very proud and he said, "Oh, I was listening to your presentation, and I give my patients a very high dosage of Xenolytics right from the start." And I say, I was really scared when I heard that. And so what you want to do is you want to distinguish between two different processes that can happen. So in principle, we are looking at senescent cells. We explained that already in the last stream. Senescent cells are cells that can also occur when you get older. So they're at the end of their life cycle, but they don't die. They stay around and turn into a zombie-like state. That means they cannot reproduce anymore and they cannot die. And so they're kind of switched off in their normal cell cycle. And they're also inflammatory. So that what you would call in humans inflammaging. But you also have a virus-induced senescence, and then of course this happens right away. It's a protection mechanism by pathogens to avoid being degraded by the internal autophagy mechanisms within the cell that can become dangerous for the pathogen, or also to be invisible kind of for the immune system. And so the so-called Xenolytics are molecules that help the organism to break down these cells, and then they get replaced. But you also have sometimes the same molecules, just with a different dosage, and some other well-known xenomorphics. And a xenomorphic will take cells that can be repaired and bring them back from that zombie state into a normal, healthy state. So they start to reproduce again, autophagy works, all the inflammatory signaling goes away. And that is what I wanted to explain here. You have to do both. And now you guess what you have to do first, if you want to do something about this problem. So yesterday we have explained that not all cells that are senescent contain spike. Not all are the rotten apples in the middle with the worms inside. You have bystander cells that become senescent because of the affected cells where viral debris is inside. And these bystander cells can be saved. And for those, you want to have a week or two, a time frame of mainly xenomorphics in order to bring them back into a healthy state. Yeah, so if I made this, that's why we have these apples here. Okay, so this is before any infection or transfection, all healthy red apples, and then you expose them to SARS-CoV-2 and or the injection, and then you have this picture here. In the middle, you have these rotten apples with the worms inside, resembling spike or viral debris or whatever. And then around it, the greenish apples now, they are what we would say bystander senescent, kind of infected by the rotten apples in the middle. But we can save them. So on the next slide, you see what happens when you now apply xenomorphics to this. And here on the right picture, you see now these green apple turn back into a healthy state. And now what you're left with are the rotten ones in the middle. And now you come in with the artillery, with the Xenolytics. And then on the next slide, you see what happens then. So then you will selectively clear out these cells that do contain the viral debris, spike protein, and other things. So that is basically what we try to do.
This is a very beautiful concept. So in effect, it's kind of like when I think of stroke disease, when we think of what we call the penumbra, the area around the stroke that is still potentially viable, but has not died yet. And everything that you're trying to do is to try and get enough oxygen and blood supply to this penumbra so that these cells will come back to life and improve the outcome after the stroke.
Mm-hmm.
And it's very similar to what you're saying here. So, okay, so let's look in some more detail then of your xenomorphics.
Yeah, so the xenomorphics, I can't show all the molecules here today, and I cannot also disclose all the little secrets we have. So, but one that is commonly used by us in many of the liquid products is rutin. So rutin is well known as a phenolic compound that can do a lot in regards with spike protein, with inflammation, with neurological problems, and so on. It's a very useful molecule, but one of its main properties is actually it's a good xenomorphic agent, and you can see that there on the right on that chart. You see SASP signature. That's a secretory cytokine profile that the cells, you can measure when the cell is very senescent, and then they excrete these SASP molecules, and that is where you can measure how senescent that cell is. Is one of the markers, and you can see that rutin was very good at different levels to bring down the senescent state in these cells at a pretty low concentration even. So actually with the xenomorphics, you want to be really low concentration, good bioavailability, but low concentration. And so you want to help these cells. The organism can rebuild these cells when it has access to these kind of molecules. There are many xenomorphics, but I'm just describing some here. Luteolin also, that's in one of the products. It's inhibiting senescence by ramping or modulating SIRT1, sirtuin-1, and p53. p53, as we know, is dysregulated by spike protein. So we have done a lot of research on p53 and how to re-establish p53 expression. And so in this case here, one of the documented molecules that can do something like that is luteolin. So it's an excellent xenomorphic in that case. It has shown here to reverse senescence at a very low dosage, 2 micromolar.
I'll add one thing here. Is that straight away as I saw that, I realized that the P53 is one of the critical molecules with regards to preventing cells turning cancerous. So one of the interesting side effects of what you're doing here, if this is bringing benefit, will be to reduce that risk of cancer broadly in the host. Very interesting. Okay, let's go on here. So are these still more xenomorphics?
Still a xenomorphic. There was this Japanese study, and they couldn't restore bone volume because the stem cells were senescent, and then they used the green tree extract epigallocatechin gallate, and it could restore the bone volume by reducing the senescence in these stem cells. And that is exactly what you're looking for. That's what you need to do in order, because if you look at these rotten apples, it also happens in our stem cells, and we have to be very careful. So the main reason why you want to use the xenomorphics first, before you do anything else, is to protect your crown jewels, neurons, myocardial tissue, stem cells, especially in the bone marrow, and progenitor cells. Because, and like I said also in the last interview, your ovaries and the reproductive organs. So there you want to rescue as many as possible, because these are our crown jewels, so to speak.
So just a quick recap so people understand this. Effectively, what Joachim was saying is that you have two types of cells. These ones that are going to die and are of no value, but around them in the green are some cells that you can bring back to life. You need the xenomorphics to try and impact on these before you try and destroy these, because if you gave the Xenolytics, it would destroy all of them, which would then make you lose good, potentially productive cells. So when you use the Xenomorphics, you end up with a situation where you have these cells that were green now becoming red and leaving only the rotten apples, which when you then use a Xenolytic, it will remove them. About right?
Perfect. It's like a hostage situation.
Yes, yes, yes. I like that. All right, let's move on to the Xenolytics now.
Yeah. So
Okay, so we understand the Xenomorphics. Let's now say the Xenolytics. Now we want to destroy them. What are you doing here?
yeah, so the Xenolytics, this is very well known from anti-aging and from other research, anti-inflammatory treatments or regimes and dietary supplements. So you see that this was just a study here, a paper that you can see that the brains of the patient have an increased accumulation of senescent cells. And what they did, they tried senolytic agents in different ones, even pharmaceuticals on these cells, but they had different kind of viral debris in each cell. Yeah, so you see here on the right in that graphic, you see RD, RP, spike, envelope, and NC nucleocapsid protein being in these cells, and then the red bar shows you these cells that they didn't have as a control. They didn't have these viral debris in there. And as soon as you apply the different senolytics, you see the right three little, the very low bars. That is how many cells were then cleared out because they contained viral debris and or spike protein. And that is kind of like, I don't know, it's like if you have a surface-to-air missile station switching on their radar. So these cells kind of are signaling, we are here. Can you send some ordnance? That's what's happening there. It's selectively destroying cells that contain viral debris.
I see.
Good. Yeah.
Very good. Okay. Similar here.
Yeah, this is it's just one of many examples. Remember, we were talking about the lymphatic system and astrocytes being affected, and they are as they're sitting right behind the blood-brain barrier, and they are heavily hit. They tried here with what happens when you apply senolytics to astrocytes that were infected or transfected with viral SARS-CoV-2 viral debris, and works excellent. You can then, one of the markers we use or we recommend doctors to use in patients, the so-called GFAP, which is in this case measured and then goes down. So that is a very clear marker for astrocyte inflammation or senescence in this case.
Mm-hmm. Excellent. All right. And irreversibly senescent cell conglomerates. These are the syncytia. Now the cells that have come together to form one big giant cell with multiple nuclei.
Yeah, they are irreversibly damaged anyhow. And like I said also in the last interview, if you look at the fried egg on the right side, there will be no chicken hatching from that. So yes, you can, without any problem, you can remove those. And so there is some proof of concept already on the next slide to see that they can also be removed with certain compounds. I don't disclose everything, but it looks pretty good. It will never be 100% solution. Look, nothing will be 100% in this disease. But if you have a significant impact on the values that and on the spike levels and on other parameters, then you have already achieved a lot. It will take us more years to come really to maybe finalize all the problem mitigation, but let's be happy with what we can already achieve.
I think that one important thing to be said with with what you're targeting is very similar to what happens with antivirals. Say when you, if you did catch a patient, not necessarily with COVID, with any infection, and you gave them antivirals early, the antivirals don't wipe out the virus. They just slow the virus replication long enough that the immune system is able to mop it up without being overwhelmed. And so in the same way, your system seems to be buying time to strengthen the immune system and allow it to then clear up and mop up the stuff that needs to be done. Would that be about right?
Yes, and also it will restore some of the immune functions. We talked about it yesterday. Persistent spike in the upper respiratory tract and in the nose will suppress type 1 interferon. It will cause dysbiosis in the bacterial, in the biome, in the nose and oral cavities. So it makes us more susceptible also for any infection. And if that's combined with spike protein persistence in the PBMCs, then of course this is resembling already an immunodeficiency state. And so by taking that out, will probably not per se restore or function 100%, but will give the organism an opportunity to rebuild resilience. And I hope that this is also happening and that we can see that now in the long-term outcomes.
Yes, excellent. So, okay, so now you have got a situation where you're targeting cells, xenomorphics, xenolytics. Where is the binding of the spike protein in this picture?
No, this is just the title. Okay. I just wanted to show a nice picture. So there is a lot of research we have done from day one of the pandemic, trying to find, because even as an antiviral, which we initially were starting out at, it makes sense to stop the virus as early as possible and diffuse and neutralize, especially the spike protein, because that is what is binding to our cells. So even if you have a normal SARS-CoV-2 infection, it does make a lot of sense to throw in a lot of binders that will now bind to the spike protein and render it pretty useless. So many of these compounds we are using, I will not disclose everything, but you can see there's different binding affinities. Anything that is blue is a good against spike protein. You can see some of the champions, of course, has pyridine, for example, but also cannabinoids. So I don't know what to say about that for people, but okay, maybe very popular in Jamaica. We see other phenolic compounds. So there are a lot of reported compounds that can block the spike protein, or let's say bind to the spike protein and henceforth block the RBD and other areas of the spike where it can fuse.
What is the brown line about?
This is the MPRO. This is a different protease. That is all you need that when the virus is intracellular replicating. It's the main protease, which is actually also a pretty good drug target, which we took even on before the spike protein in our development. And it's the main protease because it has been conserved around in all generation of all different coronaviruses. So that is a very valid drug target to use. Until recently, we talked about in the last stream that it has been put into a selective pressure now,
Yes.
and has mutated, unfortunately. So again, this is making a case for a multi-pronged approach from different sides. Don't never rely with this bastard on only 1-2 pathways. It's very agile, fast mutating, and makes it difficult to tame it because it is so inventive, so to speak, in what we get to that. We'll have a look at the next slide. You see, so these are just compounds that have shown to bind to the receptor binding domain and other parts of the spike protein. And actually, some of those are also, we use them as senolytics and binders at the same time, because you want to make sure that the binders are very close to the point of action. So you want to make sure that the availability of the binders in the body region where now cells are breaking open is there, because otherwise it would be useless. So you cannot, for example, you cannot now mix pharmaceuticals with nutraceuticals or phenolic compounds because their half-life in the blood is different and their distribution is different. So you want to use, and my favorite are the ones that break up the cells and bind at the same time. Makes sense.
Yeah, I think that as I think about it, because you've said something important that I'm not sure if it has come across, is that your first phase, xenomorphics, to give good cells a chance to heal.
Yeah.
Then you're using the xenolytics to destroy the zombie-like cells with spike protein in it.
Yeah.
When these cells die, they are going to release spike protein, which we know is very sticky, And so therefore you're using spike binding compounds to then pick up these release spike proteins and get them out of the body. Is that about right?
It's perfectly put it. I forgot to really explain that. You did that very, very good. Thank you for that.
Okay, so yeah, so here we have, this is another binder then that you use in your combination.
Yes, we brought it down a little bit. There were discussions about high blood pressure. This is actually one of the main compounds in licorice, but licorice has so many other advantages that I was reluctant to drop it entirely. It's very low dosage now, so it can't really cause any trouble in regards of high blood pressure, but it's still there to bind, for example, spike protein. Yeah, so that has been shown here very clearly, very efficiently. That's a good part.
And since you mentioned the high blood pressure, I'll clarify what we had said. A lot of people, I suspect, may be asymptomatic with regards to spike. And so the question I was asking, is asymptomatic presence of spike similar to hypertension or high blood pressure? It's very silent, but even though it's silent, it's still doing damage to the body. And it therefore is critical as part of your longer term health strategy to identify as many things that can cause harms. Almost similar, we don't wait for people to get full-blown cancer. We try and screen them to identify and deal with it early. Are you thinking along a similar line with regards to these products?
Yeah, look, it is very well documented what spike protein can cause. And it's a array of several hundred different signaling pathways that are off. And so it's always good to get it out. And of course, people are in a different state of health and resilience. And you have a lot of people that don't have any symptoms and they're fine even in the presence of spike, yes. But first of all, it's a cumulative process. Every new injection, every new infection also is adding now to the already existing load. The body, we find spike proteins, it's very difficult to really nail it down. How long has that been now persisting? But it seems like 2 years, 2.5, 3 years, it can persist in cells. So that's a long time. And statistically, we all get COVID every 2 years, more or less, across the board. Mm-hmm. So that means you will always replenish the existing reservoirs. So I don't know. I mean, if somebody feels fine, he probably doesn't have to do anything. Everybody is his own captain in life. But if you are symptomatic, so now the main point actually is to give doctors and therapists something at hand now to test first and see if the patient that is now developing a chronic illness, if that is fueled, caused, or even triggered by spike protein or the vaccine or the virus.
Yeah,
because it's hard to distinguish. Yeah.
So when we look at the gut here now, and I'm thinking carefully about what you said before, xenomorphic, xenolytic, then spike binding. Why is the gut after that? Wouldn't it have been logical to start with the gut first?
Can do that after. Yeah, you can do that. You have to do that actually in parallel, because if you look on the left, you see two different slides. One slide is showing the bacteria producing spike and virus, breeding viruses, and on the bottom corner left, you see the gut epithelial cells being full of virus and spike protein over a very prolonged time. So now this is one of the few places where we have a conundrum, because we have two different host cells harboring virus and spike, and not one intervention will clear both. Yeah? So you need to go with the Xenolytics, for example, for the epithelial cells, and you need to go with, until now only documented, antibiotics. We're working on other solutions into the gut and clear out both reservoirs at the same time. That's a tricky situation, because if you don't do a good job on one side, be it on the bacterial side or on epithelium side, you are risking a reinfection, a cross ping pong infection between these two reservoirs.
Yeah. Okay. Yeah. So as I'm thinking now, because the gut has always been, in my view, a huge issue from very early on. It's starting to get more attention now. How One of the important things is that when we say bacteria, I remember Carlo Brognia's work, and it does seem that it's not the bacteria in whole, but very specific bacteria like the Clostridia species. And if these are kept in check, you then have a better chance of resolving it. And now, in my view, the endothelium, the problem is as you said, if it's whether or not it is in a senescent state, because the endothelium normally replicates every 5 days. It dies and goes on, but it seems as though some of these cells are just not dying, and so therefore, if they've got spike in them, they are stuck in that locked state, and so therefore you have to kill them. That's where the Xenolytics come in. Um, at the same time, else they will reaccumulate more spike.
Absolutely. So that's what we are seeing some doctors now taking this route very successfully. So we're waiting for more data. And so on the next slide, I can show you what we are still doing on top of all that, because again, if you do antibiotic treatment combined with senolytic treatment in the gut, you will liberate a lot of spike and viruses. And so we have found another very useful compound. It's a humic acid. It is a pure high molecular, pure humic acid without any fulvic content. And that has shown in some trials here that it can encapsulate the spike and even the whole virus in working almost like a hydrogel, like a chelator in the gut. And the doctors using these kind of compounds are very happy with their patients, with the overall also markers that they can obtain from the gut health and gut dysbiosis and gut inflammation. So that seems to be a very good idea to add that next to the antibiotics and senolytics at the same time.
I must admit, when I heard about humic acid, I knew immediately this was exceptionally important. And the closest I think that could do something similar would be activated charcoal. Whether or not it would do a similar binding, but it's not a pleasant thing to drink. It carries, I think it can cause a few more gut issues. So humic acid, if it is as clean as it is, that would be an incredibly valuable tool in this whole process. Okay, let's shift on to what is this? Receptors, UND fusion enzymes. What are you talking about here?
Sometimes I have German slides in between. Excuse me.
Oh, and okay, I understand.
This is how, look, again, we can block the spike. We can maybe do something about intracellular replication of the virus, but also we have seen that before. The virus can not only bind to ACE2, it can bind to over 20 plus main or co-receptors that it can fuse with the membrane of the cells it wants to enter. So it does make a lot of sense to use molecules that can bind these receptors. That was the work of the first 2.5 years for us to do this, because in the beginning everybody was just saying, only ACE2 and that's it. And it's not true. It's now well documented, even very strange ones like DPP4, that's a MERS receptor, or even some HIV receptors and so on. So it's a very crafty little pathogen here. And so you want to block as many as you can, so that you block the spike and you block the receptor, so it cannot, you block the key and you block the lock.
Yeah, it's important to understand, and this is not being conspiratorial, but I would think that at this point, if someone says that it's quite possible that these kind of changes were engineered, it highlights the importance of how complex it is to be able to unpick this lock. This is almost like now a bank vault that you're trying to crack, because it is so sophisticated when you look at all of the entry and interactions that this virus seems to have.
Yeah, if you really go through the whole research that is available, and you will have moments of awe, where you are in awe of this bastard, where you say, like, how is possible that something that sophisticated and that incapacitating and crafty has been, or was accidentally released or whatever. I don't care how it came about. We just try to stop this.
Yeah. So let's look at this AXL. What is this? And again, you're trying to block the receptor, the ACE2 receptor here.
Yes, AXL is another receptor, like a alternative to ACE2. Now you see it's almost as efficient in fusing with the spike as ACE2. And we have a university in Sydney, Australia, and they are experimenting with some of these products, and they did that's what you see on the right side. They did a lung carcinoma trial in vitro with these liquids, and were very happy with it. And so one of the effects was the AXL expression was significantly downregulated by a very low dosage of this liquid. So it's just one example that we are always happy when we get some confirmation of a combination of dietary supplements in that liquid that can do what we before hypothetically were hoping it will do. Yeah? So that then I'm always very happy.
Excellent. And so the furin cleavage site, without the furin cleavage site on the spike protein, there would be no pandemic. This this is a huge point. And what do you mean?
And look, it's this cleavage that makes the spike protein so dangerous, because it now can cleave and separate the S1 subunit, which is the most dangerous part of the virus. So it makes it orders of magnitude more infectious. And so it was the main gain of function done to that spike protein. And that is, I think, if you don't enter into conspiracy theory, because now a lot of governmental agencies, including some intelligence services, have clearly now stated that this is probably a man-made chimeric construct. And so the furin cleavage site was the main advancement in their research of making that thing possible. And I don't see much out there, people trying to talk about furin inhibitors. I mean, it makes a lot of sense. I mean, if that is what makes that thing so infectious. So we were very early on. I was being brought aware by some excellent scientists that were already looking at the gain of function. That told me, hey, you have to look at the furin cleavage site. So you see that we are using a lot of furin inhibitors.
Yeah. Excellent. And again, now it induces heme1 oxygenase 1. And in this setting here, what is the relevance of the spike protein inducing this enzyme?
What happens is, because it does that, it kind of also causes syncytia formation in the liver and the kidney. Sorry. So that is one point. When you try to get spike out of the system, you want to have it neutralized, and you want to be careful that you, in the process, don't do any damage while it gets excreted. And so quercetin, for example, shows here very good efficacy in preventing this syncytia formation in the kidneys. That's why I put it there. And so it's only one of the countermeasures you can do. Also, the furin inhibitors, for example, the furin inhibitor. Excuse me.
Yeah, go ahead.
Oh, I can't see you, so no problem. So the furin inhibitors are also stopping syncytia formation. So that's why I was showing that. So this is really important in order to prevent widespread organ damage in general.
And in terms of the syncytia, though, wouldn't that be more relevant in the context of an infection rather than where the patient or the individual has been stabilized now with already syncytia in place? It's not that quercetin is a xenolytic per se. It's just it reduces the ongoing formation of syncytia.
It is exactly. It's a protection against that will prevent and block the fusion of cell membranes with each other, which is caused by mainly by the furin cleavage site. Actually, that is the main problem.
Okay, so now before we step on to some of the results that you guys have been getting, I just wanted to say something in the context of what I'm understanding. You have a number of approaches with xenomorphics, xenolytics, spike binding, furin targeting, to try and prevent even with quercetin and the heme oxygenase. This is now an alphabet in terms of an approach, A, B, C, D, E. And one of the things that I've learned about patients is that you can't necessarily do A, B, C, D, E on everybody. Some people need E, B, C, A, D, if that makes any sense. How does this fit in that kind of nuance that we see with patients?
Yeah, we are not talking about disease expressions that have developed. We're talking about a persistent pathogen that is nested in the cells. So you can go in a different following order, but then you're running so if you provide the binders and then later the Xenolytics and the Xenomorphics at last, you will do a lot of damage. You will not have these results, most likely, I think.
And so therefore, in effect, that sequence that you do, Xenomorphics, Xenolytics, spike binding, furin inhibiting, is the most optimal sequence in order to get results.
As far as we know. We are still at the beginning. We have only preliminary data that is more of a proof of concept than already now results that you want to scream from the rooftops. But it's going in the right direction, and so we will learn. There will be maybe other components be added. I'm not sure yet. We have not even understood the whole scope of the problem. So how can we be confident in saying, "Oh, we have it solved," like other people do? That is not very scientific.
Yeah. Excellent. So, in effect, then, what was it that you saw here?
Yeah, this was doctors that were using these exactly this following order. Actually, they only used one product, and they just started slow, and then ramped up the dosage over the course of time, and it was like, I think it was 8 weeks in this case here with 2 cases. And they were just measuring with the laboratory from Professor Koenig in Magdeburg. They were measuring or quantifying the persistence of spike protein in different regions of cells. So you could see that there was over 90% reduction in the course of this time frame. And so we find that pretty exceptional, because to my knowledge, nobody has ever published anything now in the scientific literature, anything like that.
Wow. So that's quite fascinating to be getting results like that. But again, your critics would say that's only a few patients. We know that sometimes it doesn't necessarily scale so easily. But as you said, this is a trend in the right direction. So in this case here.
Yeah, so you see in July 24, that was the initial diagnostic. You see that this patient X, we call him, has a spike protein in the immune cells, in the PBMCs. So plasma, serum, and exosomes were negative. And the PBMCs, the immune cells, were positive with 188 picogram, which is considered high. So the level of detection starts at 4 picogram. let's say anything between 50 and 70 is moderate, medium, and everything above 100, 150 is already considered very high. It can go up to 1,000, but that is exceptionally rare. And then you see after 2 months, there was a second diagnostic done, and you see that it stayed negative, of course, in plasma serum exosome, and the spike in the immune cells went down already by 90% to 17.28 picogram from 188. So that is significant, I would say. And then another month later, they continued. It was down to 5.18, which is barely above detection threshold, which is a level of detection is 4 picogram, as I said.
And would you say that the symptoms improved in this patient X?
Yes, that's what is reported from the treating doctor, that the neurological symptoms improved. Yes.
Okay, all right. And let's see patient Y here.
Yeah, so there was a female patient, and that is a different picture here. You see, it in this case, it was negative in plasma serum, negative in the immune cells, but positive in the exosomes. And you see here now the variety of different data coming in. And so again, this was a 2 months treatment, and the spike in the exosomes went down from 228 picogram to 14.25. So that is also significant, I would say. And I think the patient is still or was on treatment, and we will get some follow-up data. I'm sorry, I lost that a little bit out of sight here. We have a lot of things ongoing. But funny enough, you see here, spike protein in the immune cells was before initially was negative and then was positive after 2 months. So that is a sign that we most likely were breaking up cells that were pumping out the exosomes with a spike, and that when they go into apoptosis or into xenolysis, the phagocytes come and try to clear the debris. And when they do that, then sometimes they are ingesting some of the spike, and that's what we could see there.
Now,
Also,
when I look at the time frames there, we're talking about months of treatment.
yeah, 2 months.
2 months. A lot of people want quick outcomes. You know, they want to know you give them a week of therapy and they're better. What do you say to these people?
I can't say anything. Look, it takes the time it takes. What can we do? Yeah. So I see also some clinics and doctors, they try to accelerate this process, and I can only caution, you have to give your organism time, because, for example, when you have the PBMCs full with spike, when you clear them, okay, it takes around 2 weeks for them to be replaced. But at the same time, you still have problems in the thyme, you have problems in the progenitor cells, and you have problems in the bone marrow with the stem cells. So I would not rush it. I would be very happy if I can get this out at all to start with, yeah, and not look on the date. I'm in a hurry. It doesn't work like that. You have to give your organism time to rebuild the cells that were taken out, and to bring down the inflammation and get back into a healthy state. So I would not rush it. No, I don't think that's a good idea.
And I think that the more I think about it, especially as you said, the unique characteristics of the spike protein, this is almost more like chemotherapy. It would be kind of like somebody wanting to resolve cancer with chemotherapy within a week. It takes a long time and a number of hits to gradually remove those damaged cells and allow healthy cells to come back. I usually say to people, this is a marathon, it's not a sprint. And if you try and sprint it, you're going to do harm in the long run.
Depends where you start. You know, that you have people that don't have such a tremendous load, they are not really that ill. Maybe they can get it done in 4 to 6 weeks, most of it. And then there are others that maybe take 3 or 4 months or even longer. Yeah. And sometimes we see problems, and usually it's reinfection during that process. that people get reinfected, and then you go back to square one, kind of. And so that's also, you have to consider that. And so I wouldn't be in a rush. I mean, I can understand that people want to get better. Anyhow, taking out spike protein will not necessarily, it's not necessarily a therapy. It is what you need to do before you start the therapy. Because while you still have spike present, it's pretty futile to try to start a therapeutic intervention.
Yeah. So when we look here, this is again the same patient, X and Y, significant fall off in spike protein. And as you said, you can't guarantee that the person is now back to normal, but they're moving, they're trending in the right direction.
Exactly.
Yeah. Okay, good. And this here, what is this about?
This is a similar case. It's so far unpublished. We're trying to get it published. So it's a patient report. It's unofficial data, raw data. You see that that patient was completely asymptomatic. He had some spike in the exosomes, 60 picogram, which is pretty low, but he had the incredible load of 1,000 picogram in the immune cells, asymptomatic, really credible. And then after 6 weeks of taking, and he was pushing it, he was taking all the other extra supplements, he went down to 10 picogram, which is a 99% reduction in 6 weeks. So I find this quite remarkable. From I hope we keep getting data like this.
Yeah, no, that is critical. And so now the final section almost is to do with this microclotting, which seems to be a big issue with regards to long COVID, post-COVID. And again, I'm one of those things that I try and highlight to people is that when you use the term long COVID, a lot of people can get distracted because it feels too broad. And I sometimes think post-COVID is better because it is such a range of symptoms that people have. So let's look at what you got with regards to microclotting. What is this about?
And it was just like a collage to make it stick. Okay, when you speak to any of these laboratories, there are only 3 actually that are really doing that on a regular basis for patients. They all say the same. They say the micro clotting that you can visualize with amyloid staining in the blood samples of patients is so prevalent that they have a hard time to find blood without it. So the percentage of the population walking around with microclots is extremely high. And so I think that it goes pretty much hand in hand with the persistence of spike protein, especially in the endothelium parasites and in other regions where you are kind of triggering a constant amyloid aggregation and a constant clotting cascade. So I think that these are the 2 main drivers, in my opinion, that keep this whole chronic illness wave going.
I think it's important for people to understand that with the microclots and the microclotting, it's not that it never occurred before. And these clots they're not normal little clots. They can't be dissolved easily with any of the standard dissolving agents. And so if they get big enough, and this is when I spoke with Professor Kell recently, they are finding that the characteristics of these clots are amyloid. So the microclots are amyloid, but when they get bigger, they are also amyloid as well. So they found in Liverpool that when they took out the clots that were for thrombectomy, they had this same amyloid pattern. And so this is pretty serious stuff. It seems to be that microclotting is a precursor to bigger clots, but it's not clear the link.
Yeah, we are looking at that since a long time. We do have some hypotheses, and you will have Kevin next week on the conference. He can give you more insights. We are collaborating with that laboratory there in Japan, and so we did receive samples of these larger clots. At the moment, I'm again having some objections here. We're looking at a post-mortem sample. And so post-mortem, you have certain parameters that are changing, pH, temperature, and others. And so I would prefer if we get our hands on thrombi that are coming out of a living person, and we can analyze those. That will give us better data.
Yeah,
that's
excellent.
the ambiguity I see there.
So in terms of this here, you guys found, again, because of this microclotting, a significant reduction in the numbers. What is this slide showing here?
Yeah, this is just to make it visible. This is a laboratory analysis in Switzerland. It's the Inus lab in Zurich. They have a very good methodology in the laboratory to detect, quantify, and look at the size, distribution, and composition of amyloid microclots in the blood of especially long COVID patients. And the left slide was before the patient was starting to take some dietary supplements, similar, same clinic as where we had the spike results, and not the same patient though, but the same regime. And then you see after 8 weeks on the right slide, I would say quite significant reduction in the number and sizes of the microclots. So that was also documented on the next slides. You see also the complete documentation about different levels of amyloid, what kind of amyloid. They are very throughout in this laboratory. I can only recommend anybody that's in Europe and has the possibility to send blood for diagnostics there. This is the best I've seen so far from the analytic and methodology and the accuracy of what they provide. Again, you see before and after levels, and you can really compare all different parameters, and it gives you a very good situation assessment of what is happening in that patient in regards to microclots at this given time.
Now, here's an important question, because you've done the test before, you've done the test after, say, about 8 weeks of intervention. What would happen if you then did the test again 8 weeks later, having stopped it? Would you then see a recurrence of microclots at the level that we had before?
We will try. We will have a look. Of course, that could happen theoretically. But yeah, if you ask my opinion, if these two things develop in the same direction with the same intervention, it's most likely that it doesn't reoccur that quickly, and unless you are exposed again to the spike protein. It's the driver. Since
and
spike,
so this is just showing they fall off in terms of the same case.
yes,
just showing the.
Yeah, and look at that. It's again significant from 49 to 18, 73, 123 to 82, etc. So, and it's not like you have to be completely on zero. I mean, to have small and a few amyloid microclots circulating, as you said before, is not something completely unusual. Just the number, size, and the prevalence is now astonishing.
Yeah, I think it's important in the context to say that what the microclots are likely to be doing are blocking little capillaries or small arterioles. If that happens in a few locations like muscle, you may not notice much. But if this is happening in a brain that is already affected by other conditions, you may then start to see symptoms. So it is quite significant in the longer term. So here we have, again, similar principle.
Yeah, it's the same also before and after. Sometimes 3 months, there was not such a tight monitoring, but you see again, left side is before treatment and or intake, and the right slides show you the same patient after. And it keeps coming in like that. So we have to do follow-up, yes, you are right, and see if that reoccurs. But you also have then in the follow-up, you have to determine has the patient been reinfected or not. Yeah. It's always some ambiguity again here because people don't notice, they don't test, and actually it's difficult to really narrow it down because as we talked about yesterday, the nucleocapsid will not give you a clear indication. Maybe a spike test then would give you a better indication, because we saw yesterday when we we tried again the new samples of the quick test, that there were 2 patients working at the lab where we developed those. They had COVID a few weeks ago, and they also still were positive on the S1 subunit spike test. So that gives you an idea that it's practically after each infection also that you find S1 subunit circulating.
Yeah, I think that one of the critical questions is that, do you understand how these therapies are impacting on the microclots? Do you have any idea as to pathophysiology?
Mm-hmm. I can give you around 200.
Any main thing?
Look, I'm working on the, no, I'm not going to say that. Okay, there are anything from amyloid degradation and amyloid buildup. You have the protein C being downregulated. You have endothelial inflammation. You have high fibrin, high fibrinogen. You have high LPS. LPS can bind to spike protein. You have high serum amyloid, etc., etc. So I could go on for like a whole list where it's well documented that many of these molecules can interfere. Simply speaking, as long as there's spike in the endothelium and spike in circulation or wherever active, it will fuel many of these cascades that are contributing to the microclotting and to the thrombosis development in the patient.
Yeah, I think that one of the critical points that I would be saying is that when I'm looking at the research, and I think it's valuable that we do independent parallel research, so meaning that I am looking at different things, you're looking at different things, and it's how the research dovetails together. And what I would say is that everything so far with regards to the post-COVID pathology is about inflammation. And when you recognize that the spike protein is driving these inflammatory changes, and I think this is the important point that I'm taking away, is that if you have immune cells with persistent spike protein, they are locked in that inflammatory state. And when you then have a situation like that, you can't resolve inflammation until you get rid of them. And so I think that this is the important point. I think the only thought I have is that because my research had always pointed to the people who are most vulnerable are people who had pre-existing inflammation. I call it the embers of the fire. And because of those embers and because of that inflammatory baseline, these inflammatory cells that were there then get infected with spike protein. Okay. The problem is then, if even when we remove the spike protein, if we have not addressed that underlying ember, the pre-existing inflammatory process, what then happens is, you're quite right, on the next infection, this can do it all again. My question is that I want us to be able to remove the spike, remove the inflammatory cells, and remove the embers that were existing before.
That would require a drastic change of lifestyle in many people. Remember, we talked about that 4 years ago with Dr.
Yes.
Stefan and others. So what I did is I am right in the middle of that is an assessment of how chronic preconditions that are caused by mainly environmental factors and let's say eating very bad food and EMF radiation, just name it, air pollution, microplastics, etc. So we're looking at all that and giving it a score. And then we would say, okay, that's 2019. That was before the pandemic. That was the situation of chronic illness in the US, for example. And then this wrecking ball, SARS-CoV-2, and that spike protein was hitting that already chronically ill population. So if we are happy, I would be very happy if we just bring back the status quo before people got transfected or infected. And then we can start to work on what made them sick in the first place. It's a very easy rule of thumb. Anybody having chronic illnesses, especially diabetes 2 or dysbiosis, intestinal inflammation, obesity, they have very high levels of furin to start with. Yeah? So they are very susceptible for this spike protein and this virus. And also they have very high levels of heparinase, which is degrading their glycocalyx, which is the last protective wall against lipid nanoparticles and spike protein in your inner arteries and in your vascular system. So when the glycocalyx is damaged, then the spike can come in easy. And if you have high furin levels, then it makes it even easier, etc. And so if you have already dysbiosis and you have been eating a lot of food with pesticides and glyphosates, you have dysbiosis already, and now the virus or the spike comes in there and gives it the rest. Your tight junctions have already been compromised. Your blood-brain barrier is open from EMF radiation. So yeah, I mean, the interplay between chronic illnesses that were caused by other factors and now the spike protein is difficult to untangle. So that's what I'm hoping to do next is that when we succeed in getting the spike out, we have to do, before we do that and after, we have to do as many proteomics and epigenetic diagnosis as possible to get a good status quo before and after. Because when we succeed to get the spike out, then we will have a homework list of what is now left over to be addressed next.
Yeah, this is great stuff, Joachim. I mean, I think that there is so much to learn here, and I think that what you have achieved through your research, through the investment in terms of these products, is that you are ahead of the game. Now, it doesn't mean you've reached the finish line, but you are so far ahead with what you're doing that we have hope that there are answers for so many people who are sick. As we close out on this, Joachim, and we think about the principles, and let me just summarize it in case somebody has just come in. The premise is that we have a number of cells that are very much infected with spike protein, and in the process, these cells are needing to be destroyed. So let me give the example. I'm going to run through your slides here. And essentially, we've got here, these are the healthy apples. This is what happens or what was there before exposure. After exposure to spike protein, you have some cells here in green that represent the cells that have the capacity to regenerate and come back to normal. These cells, however, can't, and they need to be destroyed. So before you destroy these cells, what you try and do is you try and protect these cells by using xenomorphics. So this first stage is the xenomorphics, to protect these cells so that they come back to life. You're left with these rotten apples. These rotten apples are then taken out by your Xenolytics, which destroy them and remove them, and leaving you with a cohort of healthy cells. In parallel to that is the removal of the bacteria that could be the cause of ongoing circulation of spike, using products like humic acid to bind the spike protein, and sometimes antibiotics or other products to try and get rid of the bacteria that are reservoirs, and at the same time destroying the lining cells that have residual spike protein in them. The process that you would have done would have led to the fact that inflammation goes down, spike protein goes down, clotting goes down, the patient feels better, and you have brought them back to where they were, hopefully pre-pandemic. Is this about?
Yeah, thank you for the wrap up. Very good.
Yeah, so I think, yes, go ahead, Joachim. Final thoughts then.
And final thoughts are like we have to get into now. I'm negotiating now with some groups in the US to go into larger surveys and hopefully real clinical trials, and comparing also other methodologies. And I'm not per se at all criticizing any other methodology that goes into the same direction. I would highly appreciate that we go and do trials together and compare different methodologies to try to figure out where are the weaknesses and the strengths of each intervention or regime, and then maybe be able to work together, collaborate, and not be competitors and try to scratch each other's eyes out, but go and work together. So that would be my plea to everybody that's in that industry, trying to do something about it, to come in with us and gather data, compare different protocols. And I think there's room enough for everybody because each intervention does have some good sides to it. And so let's see that we can do this together.
Excellent. I'd like to say to everyone, if you are interested in having the spike testing done, which is in some cases is in the preliminary stage, please look for the link affiliated. It will be either in the description or just on your screen right now or in the comments. Look out for the links so that you can indicate to us that you are interested in further work being done and further intervention in terms of your health. Remember, your health is your responsibility. Don't think for a second that anybody is going to stand in the gap for you in this situation. There is still so much work to do, and we are just lucky that people like Joachim are doing the work for us. Thank you very much, Joachim. I'm sure we'll have further conversations in the future. Have a great evening. Just stay with me for the outro.
Thank you, everybody. Have a nice day.